As a repeated piece of advice.
So,
maybe Radu wants to say something.
Radu, do you want to say anything?
Go ahead. Looking forward to hear it.
Okay. So, my name is Eileen FitzPatrick, and I worked in Dr. Radu Kopa's lab here at PSU. And obviously, this is the defense presentation for my Master's of Biology. And today I'm going to be talking about microaerophilic fungi from the Ascomycote with a very broad pH tolerance and that harbor Proteobacteria in their hyphal cells. So, a general outline of my presentation today. First, I'm going to talk a little bit about the fungi that I studied, and we're going to have a little bit of an overview, just a quick run through of some of the topics I'm going to be talking about. Then I'll talk about the isolation and phylogeny of the fungus and some of the growth parameters that I went through and established for them. I'm then going to talk about the putative symbionts. We're going to look at some microscopic images. I'm going to talk about the experiments we do with antibiotics and the endosymbionts, the isolation and identification of the bacteria, and then finally the fluorescent in situ hybridization experiments that I did. And then I'm going to have a little discussion. We're going to try to wrap some of this up, what we've talked about with the fungus, what we've talked about with the bacteria, and acknowledgements. So the kingdom fungi has 7 phyla, which are here. There's over 1.5 million species in this kingdom, and they range from single cells to filamentous forms. So this is an example of a spore from one of my lab isolates. If this were a single cell form, so think yeast, these would bud, this budding cell would come off and you would end up with 2 daughter cells, just like you would in bacteria. This is the filamentous form here. This is the other one of my lab isolates. And you'll see that the cells have stayed together and they're forming this hyphae, which when it looks like this is referred to as a mycelium. Come on. So fungi's role in the environment is actually pretty broad, but primarily one of the key facts is the fact that they're osmotrophic. So they actually excrete enzymes and acids into their environment that break down the macromolecules, and then they absorb the smaller constituents, and that's their feeding style. Because of this, they're actually the primary degraders of all biomass in terrestrial environments. And it's estimated that over 90% of all land plants are actually involved in a mycorrhizal relationship with fungi, and these relationships are integral to plant health and development. So because they do excrete these organic acids, one of the things that they're involved in is bioweathering of minerals and glass. So they'll colonize silicate minerals, so think quartz and olivine, composite minerals like glass, and iron oxides. So they actually have seen preferential colonization on minerals and materials that contain iron. The upper picture here is a quartz crystal. The black arrows are indicating what you would see in a typical weathering pattern that's not bioweathering. So this would be nicks, environmental causes. The white arrows here are indicating areas of bioweathering. So they look quite distinct and different from abiotic weathering. They also do a physical degradation, and this is specific to the fungi and to some other filamentous forms. So what you see here is an iron crystal. That's a mineral that contains iron. And these small lines here are fungal hyphae. So you'll notice that they're working their way into the micro fissures here in this material, where they will excrete acids, and actually they can swell as well, which will cause the mineral to crack and fracture. So a lot of the acids they excrete are actually low molecular weight organic acids, and there's a wide variety. So oxalate, acetic, butyric, valeric, propanoic, there's quite a range that they can excrete. And they do it for a number of reasons beyond feeding. So if they're exposed to heavy metals in their environment, so the examples I give in copper, zinc, cadmium, though there's more than that, they will excrete organic acids that will act as ligands. So what they're trying to do is surround that metal ion so that it's not interacting with them and it reduces the toxicity. They can actually just form the ligand complex. They can also, some of these organic acids can cause chelation and precipitation out of the solution as well. So mycorrhizal fungi specifically will excrete organic acids into their environment, and they do that when there's a nutrient deficiency, and those organic acids then break down amorphous phases in minerals, so they'll free that nutrient up for their use and the use by the plant that they're actually occupying. So as a side, the organic acids can also change the equilibrium of metal ions in solution. So oxalate, and that's what I'm talking about here specifically. So this is the oxalate ligand complex. So you'll see here there's 2 iron 3s and 3 oxalates. So when this complex forms, there are 6 protons that are freed. Those come from the oxalate. And that causes a drop in the pH. And that drop in pH allows the reductants that are naturally in the environment to reduce the iron 3 to iron 2, which will then be released from this complex and into the environment. So fungus have also been found to carry endosymbionts, and 2 fungi from the Microaerophilic specifically are where these have been found. So the first fungus that they found endosymbionts in the fungi was Gigaspora margarita, and this is an image of the Gigaspora margarita Hyphae. What you see here is they've stained with what's called a backlight live dead bacterial viability stain. So the red represents propidium iodide, which can cross damaged membranes, in this case the fungal nuclei because they're relatively porous, and stain the nucleic acid so that they glow red in fluorescence. The green is the bacteria, and that is the Cyto9 that is in the stain as well. Cyto9 will cross undamaged membrane and then stain the nucleic acid there green. So that's what they're trying to show here. These are fungal nuclei, and these are the symbiont, which is the Glomeribacter gigasporarum. Okay. In this relationship, this is actually an obligate relationship for the bacteria. They can't or won't grow outside of the fungal hyphae. It's also a vertical transmission, and what that means is that if the fungus is infected with this bacteria, that the bacteria then travel with the spore. So it's a mother-to-daughter transmission strategy. And this isolate is found worldwide. So in every Gigaspora margarita they've looked at, they have found this endobacterium. Rhizopus microsporus was the second one they found endosymbionts in, and this image is the Rhizopus microsporus hyphae. And in this case, they've just stained with the Cyto9. So you see that this is the bacteria that's glowing green. And they actually contain 2 possible endosymbionts, which is the Burkholderia rhizoxinica and the Burkholderia endofungorum. Though they're 2 different phylogenies here, they are from a common ancestor. So they are directly related. This relationship is non-obligate for the bacteria, which means they can culture this bacteria out of the fungal hyphae and it would grow quite well under lab conditions. But for the fungus, it's necessary to undergo vegetative reproduction. So if the fungus doesn't carry the endosymbiont, there's no vegetative reproduction happening. The transmission strategy for these is horizontal and vertical, which means if the fungus is infected, or I hate to use infected, but if the fungus is carrying the endosymbiont, the spores will also carry the endosymbiont, so it's a mother to daughter. But because the fungus can exist without the bacteria in its hyphal network, if you reintroduce the bacteria into a culture containing the fungus, it will pick up the bacteria and they will reappear in the hyphae. And this relationship, these are also found worldwide. So environmental isolates of the Rhizopus microsporus are found to contain this, but clinical isolates have never found to have it. So in our lab and the 2 isolates that I worked with, they were found growing in an acidified water solution with no added nutrient. They isolated them on whey agar plates, did just an isolation streak, and found that they had 2 colonies. So they had white colonies that they gave the lab designation of Baro1, and they had dark green colonies that they gave the lab designation of Aspo1. So they took them, they put them on the microscope, and microscope examination showed that they were fungus, and they had distinct morphological differences. So here are images of my fungus. These were actually grown in a minimal nutrient acidic liquid media with a glass cover slip as a substrate. So here is the Vero1 isolate, and here is the Aspo1 isolate. You can see that they have two distinct morphologies. This is the hyphae cell of the Vero1, tends to be a little narrower. The cells tend to be slightly longer. These are vacuoles that are forming, so both of the isolates contain vacuoles. This is the Aspo one. You'll notice that the hyphal cells here are rounder, that they have a more football shape in some regards. And you'll see that there are a number of vacuoles that are forming here, and these form both in the hyphae structure themselves and at the end of the hyphae. So we did 18S RNA gene sequencing and got a partial sequence. And these are all fungus from the Ascomycote. And here at the bottom are the Verticillium SP Vero1 and the Verticillium Aspo1, because that's the genus we determined they identified as. So the first question I was asking was, when these were isolated, the original liquid that they found them in was a pH of 2.5. So I wanted to find out how acid tolerant they actually were, because that's a very acidic condition, but I wanted to see how far I could take that. And then the reverse of that was, how alkaline could I go? How tolerant were they of alkaline conditions? So I inoculated spores into, again, the minimal nutrient media, and that's 0.5 milligrams per liter yeast extract. So typical media would be 5 grams per liter yeast extract. So we've really reduced the nutrient that we're giving them. And then we amended the media with either hydrochloric acid or sodium hydroxide. So this table shows the results of the experiments that we did with the hydrochloric acid. And what we found is that both isolates will grow down to as low as 1.2 pH. On the other side, with the alkaline tolerance and the sodium hydroxide, we found that they will continue to grow to at least a pH of 13.55. And earlier researchers have reported that this fungus seemed to go better in the presence of iron 2. When it was added into the culture media. This is a known microconstituent of borosilicate lab glass, so it's just one of the trace minerals or one of the trace elements that shows up in the borosilicate lab glass. But we were struggling because it's really hard to determine if this is a biotic oxidation of iron 2 or an abiotic oxidation of iron 2, because iron 2 is very unstable in most conditions. So here you have this graph, and that's what we're trying to show. So these lines represent various pHs. This is the concentration of iron 2 versus time. So at pH 4, what you'll notice is after 180 minutes, most of the iron 2 is still in solution. So at lower pHs, it's stabler and it lasts longer in solution, though it will eventually disappear and become iron 3. As we move up in pH, you'll notice by the time we reach pH 7.75 that the iron 2 is disappearing from the solution very, very fast. So the higher your pH goes, the less stable the iron 2 is, the less it is maintained in that solution. So this we had a handle on. We maintained the cultures at pH 2.5. So we were feeling pretty good about that. But then you go, okay, if they're extracting additional energy in the presence of iron, is it iron 2 or iron 3, and how do you tell? So we were trying to look at iron 2 and iron 3, and we knew that iron 2 to iron 3, that oxidation happens aerobically and anaerobically. But the reduction of iron 3 to iron 2 will only happen in anaerobic conditions. So now we have to control the atmosphere, because we have to force this fungus into a condition of anaerobiosis or close to anaerobiosis. Iron 3 and iron 2, if it's using iron 2, so if it's providing additional energy, it's got to be very little. Because in acidic conditions, iron 2 doesn't provide a lot of energy. So the theoretical maximum here is 120 moles of iron 2 oxidized with oxygen produces enough energy to synthesize 1 mole of glucose. So we have to control the atmosphere, and we have to be able to see what energy, additional energy might be coming from the iron 2. So we're controlling nutrient availability. So we limited the amount of organics we were giving them so that what was coming from the organics wouldn't mask what we would see from iron 2. And these are the results of that experiment. So this is oxygen concentration along the bottom, and that's as a percent of headspace. So these were in sealed jars and gassed with tank nitrogen. And that goes from, we assumed 21, this would be atmospheric, 21 to 22%, we assumed 21%, that's atmospheric oxygen, to 1.6%, which is the amount of oxygen that's inherently present in tank nitrogen. And then we measured phospholipid phosphate. And we measured phospholipid phosphate not because it was going to tell us how much fungus we had, but it was going to give us a proxy that we can compare between cultures. Because each phospholipid contains 1 phosphate. So you measure those phosphates and you know how many phospholipids you have. So we measured phospholipid, and this concentration is in micromoles. On this graph, the blue line represents the cultures that were grown without any iron present in solution. And the red line up here represents cultures that were grown with 20 millimolar iron 2. So what you'll see is between 21% and 10%, there's really no difference. So providing iron didn't make any difference at all. But when we get down to 5% and 1.6%, we started to see a difference in the amount of biomass that was being produced when we provided the cultures with iron 2. The inset here at the bottom is the same experiment, but in this case it's 1 millimolar of iron 3. The blue line on this graph is the same as the blue line on this graph, and the orange line is the iron 3. And from looking at this, we couldn't tell, it didn't seem that the iron 3 was contributing to growth in any way, and there was no distinct pattern. So we discarded iron 3 for further work because it didn't appear to make any difference to the fungus, whether it was present or not. We wanted to make sure that what we were seeing here, that this fungus would grow where there was low oxygen, was actually happening. So we inoculated the fungus. This is a soft agar tube. So this is a tube that has enough agar in it to make it a little bit firm, but not as firm as you would typically see from, say, an agar plate. And then we added resosarine as a redox indicator. So in the pink tube, this is the control, this is the resosarine, this is the Vero-1 isolate, this is the Aspo-1 isolate, and what you can see is the fungus is growing down into the media, and the media has actually gone white, which means that it is no longer, that there's no more oxidant left in the environment, which in this case is supposed to be representative of oxygen. So if they're using iron, the next question we ask is, iron 2 in solution is unstable. It's going to naturally convert over to iron 3 in time. So there are ways for this fungus to try to maintain that iron in solution. So we grew the fungal hyphae, so here is time since inoculation in days. And what that represents is actually not inoculation from spore, but we grew the mycelium in a minimal nutrient liquid media, and when they were large enough to visualize, we pulled them out, washed them to remove excess nutrient, and introduced them to a nutrient-free media. And then we tracked the evolution of organic acids, and this is your concentration in millimolar, over the course of 27 days. So what we saw with the red line here, this is acetate and the dash is the trend line, is at the beginning, acetate was not at detectable levels, but as we proceeded day 7, day 14, and day 27, you'll see that the amount of acetate in solution climbs until it reaches a concentration of almost 3.4 millimole. The green line is oxalate, and what the oxalate had a similar pattern in that it had no detectable levels at day 0, but it also maintained no detectable levels at day 7 and at day 14. But at day 21 and at day 27, it started to increase in solution to reach a concentration of almost 2.4 millimoles. So both of these acids are capable of acting as ligands for iron in solution. So we theorize that these are being produced in response to nutrient deprivation, which is what we know happens in mycorrhizal fungi. And then they were acting to hold that iron 2 in solution until it could be utilized for additional energy by the fungus. The other interesting thing about these fungus that they found when they were doing the original examination on the microscope was that both of them contain a lot of procurrent growth and a lot of vacuoles in their hyphae. So here is the Aspo1 isolate, here is the Vero1 isolate. Again, these are both cultures that were grown on glass substrates. So here at the top in the black circle, what you see is procurrent growth. And what this is the fungus will produce a cell at the end of the hyphae, and then the cell behind it will start to produce a second cell, but it pushes it through the original cell. So what you end up with is, if you look here, this is actually the hyphal cell, and then this here is a membrane enclosed space. So it's an external yet internal structure for this fungus. And then they also created vacuoles. So here you see this is a vacuole. This is a lot like what you would expect to see in a plant with the vacuole formation. It serves a lot of the same functions that you see in plants. So the vacuole is formed. It's starting to push the other cytoplasmic constituents out of the way. And here in the Vero one, you can see the vacuole and the cytoplasm that surrounds it. And in these vacuoles, they saw movement, rapid inclusions that moved very quickly. So in this image, if you look here, you can see the movement within the cells. And then this image right here is a close-up of this structure here. So these were stop motion images that I made. So I took repeated images with a still camera and then put them together as a movie. This, I actually did. A friend of mine, James Radney from the chemistry department, came in and helped, and he had a video camera. So you can see here, this is actually real time. So you can see here, 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 all throughout all of these hyphal cells are small bodies, and they're moving very rapidly. Some of the cells contain 2. But this is what they were observing. So you know that we knew that fungus were capable of carrying into symbionts. We have fungus with small, fast-moving bodies, and we wanted to see what they were made of. So were these inclusions or were these bacteria? So the first step was we stained with the live dead stain. So if you remember from the beginning of the presentation, I showed you the image of the Gigaspora margarita and the backlight stain. And this is a hyphae of the Vero 1, and at the bottom, image B is the same hyphae, so the same field of view stained with the backlight with the Cyto9 and Propydium iodide. So the red arrows here are pointing at fungal nuclei, and you can see they're spread throughout the hyphal network. And then the green arrows are pointing to the small inclusions that stained with the Cyto9 but not with the Propydium iodide, which tells us that they have an intact plasma membrane and that they can contain some type of nucleic acid. But it doesn't tell us what type of nucleic acid. It just tells us it's membrane bound and there's nucleic acid. So we moved to acridine orange, trying to maybe tease out a little bit what kind of nucleic acid we were going to be seeing. Acridine orange is a nucleic acid stain. If it stains double-stranded DNA, it fluoresces green. And that's generally interpreted as a metabolically inactive cell. For single-stranded DNA or RNA, it fluoresces red-orange. And that tends to be interpreted, or is generally interpreted, as a metabolically active cell. So if you see a lot of RNA, you're going to see a lot of red-orange. So it's making protein, it's metabolically active. This is the image of the Vero1 fungal hyphae. The red arrows are pointing to the inclusive bodies, and these are in the vacuoles that we had seen. B is the same image, only stained with acridine orange, so the same field of view. So what you'll see is the extracellular matrix around the fungus always picked up a little bit of the acridine orange and glowed faintly green. That was something that we saw consistently. But here, this red spot and these bright green spots are what we theorize to indicate fungal nuclei. So these are our fungal nuclei. Either they're compacted or they're not. The majority of the hyphae stained red-orange, so we've got a lot of single-stranded RNA, or single-stranded DNA or RNA, most likely RNA as a ribosome. And then the small bodies inside the cells themselves stained green. So that tells us that this is some kind of double-stranded DNA, though in some cases they also stained red-orange. So then we are back at the same place we were. We can't tell what kind of nucleic acid we're seeing. The other thing that we noticed with the acridine orange is that the vacuoles themselves took up little or no stain. So they didn't stain. So they do not contain a lot of any type of nucleic acid. To be sure that what we were actually staining was the inclusions and not another structure within those vacuoles, we took video and observed, and this is a video of that. So this is a fungal hyphae stained with acridine orange. These granular kind of structures are the fungal nuclei, and in this cell here you'll notice that the moving body is in fact what is staining with the acridine orange. So to hopefully tell us what type of nucleic acid we were looking at, we went to DAPI, because DAPI stains specifically DNA and it fluoresces a very bright blue. It stains RNA as well, but the stain that it takes up for RNA is very, very dim, so you normally can't visualize it. Here is a hyphae, and we actually had to fracture the hyphae to get the DAPI to go in because DAPI doesn't penetrate cells very, very well. The red arrow is showing you what we interpreted as a fungal nuclei, and the green arrows are showing small inclusions that are staining with DAPI, so they contain DNA. So now we're pretty sure it's membrane bound, it contains DNA. We're feeling pretty confident that probably what we are looking at is a bacteria. But if it's a bacteria, it should react to antibiotics. We would expect to see, if we expose it to antibiotics, that we would see a decline in number or an elimination. So we tested a number of antibiotics, and what we were looking for was we wanted an antibiotic that seemed to reduce the number of moving bodies we saw inside the cell, but at the same time wasn't going to inhibit the fungus itself. So we ran through 5 and came up with both tetracycline and erythromycin seemed to eliminate or reduce the number of moving bodies that we saw, but didn't inhibit the fungus. We then grew cells from spore with the tetracycline present, the erythromycin present, or both antibiotics present, and we visualized them on the microscope and counted the number of cells with either no movement, with one body within them that was moving, or with 2 bodies that were within them that were moving, 2 or more. And then this is actually the total of 1 or 2 or more moving bodies. So these act as percentages. So this is the percentage of cells in which we saw these different characteristics. So what you'll notice is without antibiotic, almost 80% of these cells are carrying some kind of moving body, 1 or more, mostly 1, but sometimes 2. When we exposed them to tetracycline and erythromycin, or both antibiotics, there was a reduction in the number of moving bodies that we saw, so a reduction in the number of bacteria. So this told us that this was working. These are most likely bacteria that reacted to the antibiotic, but we weren't actually able to ever totally eliminate them. And we don't know why. We don't know if because the fungus is a eukaryotic cell, that it was somehow protecting the bacteria from the antibiotic. It was metabolizing it or just acting to not allow the antibiotic into the cells. These could have been the wrong antibiotics. Maybe there is an antibiotic that would eliminate them utterly, but we never found one. So we also wanted to see, because then from the reading, I knew that the Rhizopus microsporus, the endosymbiont inside that fungus, is actually responsible for producing a hepatotoxin called rhizoxin that then the fungus excretes into the environment. So I had seen organic acid production, and I was curious to know, was the Enterobacterium actually contributing to this? Were these bacteria responsible for the production of the organic acids? Bacteria are known to produce a lot of organic acids themselves. Or could it just be involved in some of it or all of it? Or there were a lot of questions about that. So what we did was we inoculated spore into a nutrient-reduced media and tracked the concentration of organic acids. And what we expected to see was that when you expose to antibiotics, that the concentration of those organic acids would decrease in the solution. So there would be less of them. So this is the result. This is the concentration in micromoles of these varying organic acids. The gray bars represent the cultures that were exposed to tetracycline. And we chose tetracycline because neither antibiotic was better than the other. So we moved forward with the tetracycline. And then the cream-colored bars here show the cultures that were grown without antibiotic. And the error bars are a standard deviation. So this is the opposite of what we expected. So we eliminate or we reduce significantly the number of these bacteria we see in the hyphal cells, and the amount of the organic acids shoots up. So then we have to ask the question, what's actually happening here? Are the bacteria inhibiting the production of the organic acids somehow? Or are the bacteria utilizing the organic acids somehow? We didn't know, and at this point in the experiment, there was nothing we could say about this other than it was happening. You reduce the endosymbiont, you increase the production of organic acid. So we've got a bacteria, we're pretty sure about that, and it's in the fungal hyphae. So we wanted to see if we could isolate the DNA via PCR. So we used universal both bacteria and archaea primers because at this point we don't know, do we have an archaea or do we have a bacteria? So we tried both. And what we found is with the bacterial primers, we saw amplification in both species, so in both isolates. This is the control DNA, so it's just a generic bacteria Shibonella DNA. So we know that the PCR is working. And we could sequence these bands, and what we got was this identified as a Burkholderia. And this was actually a very exciting find, because both of the other endosymbionts from Rhizopus and from Gigaspora were both Burkholderias. So we felt like we're on the right track. This is a result. This is a good result. So we moved forward and we tried to isolate. So we ruptured the fungal hyphae, spun out the fungal biomass, plated the supernatant onto TSA plates with streaking for isolation, and generated colonies. So we grew bacterial colonies. We're very excited. These were mixed colonies though. So we have a large bacteria, a large rod, and then a relatively short stubby rod. I was never able to successfully isolate these from each other via streaking, and so I ended up having to do a mechanical separation. And we moved forward with the small rod because it matched the morphology that we had seen inside the fungal cells. So the isolated bacteria, we performed PCRs using the same bacterial primers, and it came back as belonging to the Brady rhizobium. So this is not what we were expecting. We were expecting a Burkholderia. did a little research on rather than just saying, oh no, contamination, start again, did a little research on the Bradyrhizobium and found they are endemic soil microbes. They're alpha proteobacteria, and they're known symbionts. So the Bradyrhizobium are involved in nitrogen fixation, nodule formation in plants, specifically legumes. So it wasn't a Burkholderia, but it was a bacteria that forms symbiotic relationships. So maybe we've got two. That's where we kind of went with this. So here is actually the phylogeny of both the Burkholderia that we were able to get as a sequence out of the fungal DNA, and here's the Bradyrhizobium isolate. In the beta protobacteria up here, the Gigaspora, Gigaspore, and the Burkholderia rhizoxanica are actually both, these are all known endosymbionts, so ours falls very well within that structure, and then the Bradyrhizobium is down here with the alpha proteobacteria. So to try and separate this out, we had tried the generic universal bacterial primers, and all we ever had was Burkholderia. So what I did was I created a Bradyrhizobium specific primer. So this was specific to my isolate. And then using the same reverse primer, so 1391 reverse, I tried to amplify the Bradyrhizobium DNA from the fungal DNA. This is the Vero one here, and we ran 2 different concentrations because we were a little worried we weren't going to see a lot of DNA. I wanted to make sure that in very small amounts I could still see amplification. So here is the Aspergillus, and you'll see that we have banding. This is the actual DNA from the isolate. This is Shevanella DNA, so it's not just priming all bacterial DNA, and the negative control. So we were able to actually sequence these amplicons, and they came back as Bradyrhizobium. That was great. We were never able to get a clean sequence from the fungus. There was always too much interference, and so we could never get a clean sequence. So I've got an isolate. I can prime it, I can see it, but I can't sequence it. So we were trying to figure out how are we going to show that we have, the Burkholderia we're pretty confident of, how are we going to show we have the Bradyrhizobium? And so we went to fluorescent in situ hybridization. So this would show us the bacteria, so we could distinguish between the two, and we could definitively show where they were in the fungal hyphae. I constructed 3 probes, so one for the fungus that was based upon areas of homology and their 18S. So it worked on both of my fungus. I created a probe for the Bradyrhizobium based on the 16S sequence, and one for the Burkholderia based on the 16S sequence. I also used a generic bacterial probe that I got from the Ard Silva probe database, and that was the non-338. I had the probes constructed, so here's actually the sequences, and each one was flagged on the 5' end with a fluorophore. So in these images, this is the micrograph of the Aspergillus 01. This is the fluorescent image of the fungal probe in the fungal hyphae, so this is the same field of view. And it probed throughout the hyphal structures, which is what we had expected. This is the generic bacterial probe, so that we can't distinguish which bacteria we're seeing, but we can say we're seeing bacteria. And what we noticed is that it bound not throughout the hyphal structure like we saw with the fungal probe, but in very specific areas, and actually in the vacuoles, which is where we saw the preponderance of movement in this particular isolate. And then this image here is the overlap of A, B, C, and D. This is the same probe set, and this is the Verticillium, or the Vero 1, sorry. Here is the light micrograph of the fungal hyphae. Here's the probe, and in this particular isolate, it was binding more in specific cells than throughout the entire mycelium. Though these are very, very fine hyphal structures, and so it was a little hard to visualize until we got into areas where the cells were a little bigger. This is again the generic bacterial probe, and it's binding quite specifically in a particular spot where the arrow is, and then this is the overlap. So we can see that where the probe is binding is distinct and different from where the bacterial probe is binding is distinct and different from where the fungal probe is binding. So this is a new probe set. So these are the probes that I generated for the Burkholderia and the Bradyrhizobium. Here is the light micrograph of the Vera1 hyphae. Here, 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 and here we see where the Burkholderia probe is binding. Here, and vaguely here, it's a little hard to see up here, you can see where the Bradyrhizobium probe is binding. And here is the overlap. So here are these two, Bradyrhizobium, Burkholderia, and down here you see the Burkholderia. So they're binding, they're in the same hyphal network, and they're distinct and different from each other. And again, this is the same probe set. So here is the fungal hyphae. Here we have the Burkholderia probe binding. Here we have the Bradyrhizobium probe binding. And here is the overlap in the same hyphal network, but distinct and different. So what we're saying is that we have two bacteria. They both exist within the same fungus. One is a Bradyrhizobium and one is a Burkholderia. So just some conclusions about what we've learned. We have these two fungi from the Ascomycote, and they exhibit a tolerance to very extreme pHs. They grow in very poor organic nutrient, and in fact they will grow with no given nutrient. They're very slow, but they'll grow. They both appear to be microaerophilic. They both produce organic acids, which they release into their environment. And in nutrient-depleted media, they grew better under low oxygen conditions in the presence of iron 2. The Endobacterium, what we can say is there are two. One is a Burkholderia, one is a Bradyrhizobium. We were able to isolate the Bradyrhizobium from the fungal hyphae and grow it in the lab. We weren't able to isolate the Burkholderia and culture it in the lab, but its DNA was present in the fungal gDNA. With fish staining, we were able to show that one hyphal network contains both of these bacteria. And we're actually not really sure what role they play, but they seem to be involved somehow in the production of organic acids. So acknowledgements, I'd like to thank Dr. Rowdy Popa via Skype, who was my advisor. My committee chair, Ken Stedman, who actually stepped up and put a lot of work into this to help me make sure that I completed this. Dr. Dirk Owada, who we all saw. And Dr. Martin Fisk, who actually he drove all the way from Corvallis to be here today for this. So thank you guys. I'd like to thank my friends, so Michelle Thompson and Samantha Chess, Brett Anderson, and Dr. James Radney, who's Jimmy Radney, who helped me do the moving images of my fungus. My family, so Joseph and Kathleen and Deanna and Lee. My lab mates, so Manar, who unfortunately could not be here today because she's now currently teaching at PCC. Wynn, hi Wynn, Amy Smith, and Tara Chestnut. And I'd also like to thank the Portland State Biology Department for allowing me to do my work. References. And we're all blind, but do you have any questions?
Yeah, so standard protocol is that the committee's not allowed to ask questions until we kick the rest of you out. So, go ahead.
Were your Bradyrhizobium isolates that you had in
I'm sorry, I can't hear you over Radu. Yes.
Okay. Were the Bradyrhizobium isolates the isolates that you were able to grow outside the fungi, were those motile like you saw in the movie?
I thought that I saw motility, and I tried several different types of flagellar stains, but I was never able to actually stain flagella. It's really hard and kind of a persnickety staining technique. Though I did think that they were modal, I couldn't actually show it.
So it's more than just Brownian motion, they seem
It's-
to be.
It's definitely not Brownian motion.
Okay.
Mhm. Yes.
Did you ever take a closer look at the exudation of the organic acids in the semi-cured bacteria and figure out how they were contributing to that?
We actually did, as once we had the bradyrhizobium isolated out of the hyphal cells, one of the things that we did do was we examined if they would grow if we gave them as nutrient like 4 of the organic acids that we saw that increased in concentration when we reduced their number in the hyphae. And we found that they will actually grow with those organic acids. Not specifically which ones they were using, but they do grow. So that would be future work, is to see which of these organic acids they're utilizing. And so yeah, we think if we eliminate them, they're utilizing them as a carbon or an energy source. And so when you reduce their number, you see an increase in acid.
So when you're staining for different nucleic acid types, we're looking at kind of DNA indicating that it was the less metabolically active state versus red representing more active. And you were looking at a cell set that looked very banded. You had a
Yes.
very red patch followed by a very green patch followed by a very red patch. Did you see any bias between active and inactive for where your symbionts were located?
Yes. So, oh, I hit a button and it turned itself off. I have no idea what button I hit. And show. So I'll go back to that image really quick. So this image.
I think it was further down.
So this was actually a very consistent pattern, at least in the Vero 1. So you always saw that you had this vacuole that wasn't uptaking a lot of stain or any stain at all. And then these bright green patches. So, and this structure was consistent. But then here are the hyphal cells that maybe are not containing as many vacuoles, and they would look like this, red and green, kind of a mottled appearance. But where this was something that we saw over and over with the fungal cells, the nuclei being pushed to the side, these vacuoles that don't contain nucleic acid but do carry bacteria. I'm gonna mute him. I think it's rude to mute your advisor. Yes.
One more. I think you probably said this and I might have missed it, but in your fish stains for the Ascomycote strain, your fish probe for the fungal cell was kind of all green,
Right.
but then the other one was kind of localized green.
Right. And
What's the reason for that?
these probes were actually, we were probing for RNA. So these are not DNA. So this is, we're probing for ribosome.
Okay.
So seeing it throughout the hyphae was something that we expected more. And if you, they were so exciting the first time, I'm gonna make you look at them again.
Yeah.
So here, you're talking about why does it look like this?
Or if you go back one.
Okay,
those are
so-
all, like the panel B, it's
Mhm.
kind of all green, but now it makes sense that if you're staining for ribosome.
Yeah, we weren't staining, we were staining for ribosome. In this, they didn't always look like this, and sometimes they did. And this is really where it's impossible to know why. Whether because I permeabilized and then mounted these on polylysine slides. So whether I damaged the cell at some point and so all of the cellular content linked out, so we're not seeing ribosome, or these cells were dead, because you will see dead cells within hyphae. So whether they were dead to begin with and they didn't stain, or they were so fine, you'll see here they are very, very fine at the edge of the mycelium, that maybe they are staining, but the fluorescence isn't really bright enough. When you get this bright fluorescence, you can't see that as a background fluorescence. I really don't know why that happened, but it did.
Makes sense.
Yeah. So,
Yes, okay, so we kick out all of the
kick out all of
important
everybody
people
else.
and all the important people to stay.
People take food.